Coffee and Health, exploring the science behind the world's favorite stimulant. Hey there, people, Jay here. Today, we embark on a journey into the fascinating world of coffee, a substance deeply ingrained in our daily lives and culture. From the comforting aroma of freshly brewed cup of coffee to the energizing boost of an afternoon tea break, coffee touches nearly every corner of our society. But beyond its ubiquitous presence, caffeine, the substance found in coffee, holds a remarkable position in the realm of pharmacology and human physiology. As one of the most widely consumed psychoactive substances globally, caffeine exerts profound effects on our bodies and minds, influencing everything from our alertness and concentration to our athletic performance and mood. In this presentation, we'll delve into the science behind caffeine, exploring its mechanism of action, potential health benefits, and consideration for its consumption. From its molecular structure to its impact on neurotransmitter system, we'll uncover the physiological intricacies that underlie caffeine's effects. So join me as we embark on this journey of discovery, unraveling the mysteries and marvels of caffeine and its impact on human health. Whether you are a coffee aficionado, tea enthusiast, or simply curious about the science behind your morning pick me up, this presentation aims to inform, inspire, and spark meaningful discussion about caffeine's place in our life. Let's dive in. Absorption and metabolism of caffeine. The chemical name of caffeine is methylxanthine 137 trimethylxanthine. After ingestion, caffeine gets completely absorbed in around 45 minutes. It also has the potential to cross the blood-brain barrier. Predominantly, the caffeine molecules gets metabolized by cytochrome 450 subfamily CYP1A2. Caffeine can metabolized into two different metabolites. One is theobromine, or and second one is paraxanthine. Paraxanthine is the dominant or predominant metabolite, while theobromine uh, conversion is minimal. Caffeine gets converted into theobromine with the help of methylxanthine N1 demethylase, which in turn, with the help of methylxanthine N3 demethylase, gets converted into methylxanthine. And through the action of methylxanthine N1 demethylase, the 7-methylxanthine can get converted into paraxanthine, or the caffeine molecule itself directly gets converted into paraxanthine with the help of methylxanthine N3 dimethylase and cytochrome 450 subfamily 8. Now, the paraxanthine molecule can get converted into two different metabolites. First one is 1,7-dimethyluric acid through the action of xanthine dehydrogenase and A2 and A6 subfamily. The second one is 1-methylxanthine through the action of paraxanthine demethylase and subfamily A2. This 1-methylxanthine gets converted into 1-methyluric acid through the action of xanthine dehydrogenase oxidase. Both 1,7-dimethyluric acid and 1-methyluric acid gets secreted through urine. Now let's look at some of the factors that affect caffeine metabolism. So as we know, we do not allow toddlers or newborns to drink coffee because the levels of CYP1A2 is in very low levels, and studies have shown the half-life of caffeine molecule is around 80 hours. It can vary to 130 hours also. Studies published has also shown that smoking increases the caffeine metabolism. So let's see how. So we know smoke contains number of polycyclic aromatic amines like benzoylpyrene, benzo-K-fluorethene or benzo-P-fluorethene. These PHs or polycyclic aromatic hydrocarbons acts as ligand for aromatic hydrocarbon receptors, specifically in hepatocytes. This complex of ligand and AHR binds to the XRE or the xenobiotic reactive elements, which are located within the promoter regions of uh, some genes, specifically the promoter region of CYP1A2. The binding of the ligand and HR complex onto XRE recruits a number of cofactors like ARNT or aromatic hydrocarbon receptor nuclear translocator. And with the help of this, the CYP1A2 gene production or the gene replication and protein expression increases. Pregnancy decreases the caffeine metabolism through the hormonal influence of progesterone, estrogen on the hepatic enzyme, or it could be because of the placental transfer of caffeine molecules. Genetics also play a role in the caffeine metabolism. Studies have shown that a variant of CYP1A2, or specifically people with this variant, has the increased plasma caffeine levels and the low paraxanthine and caffeine ratio. It is also shown that people with this variant of CYP1A2 has the decreased caffeine habitual intake. Right. Medications like ciprofloxacin inhibits the CYP1A2, and cardiovascular drugs like propanol also inhibits CYP1A2, decreasing the overall caffeine metabolism.
Alright, let's talk about effects of caffeine on cognitive functions. As we can see, the adenosine molecule is very similar in structure to the caffeine molecule. What happens is, when adenosine gets accumulated in brain, it inhibits arousal and increases drowsiness. However, because of structural similarity, caffeine antagonizes the binding of adenosine to the receptors, which does not allow us to fall asleep, which increases alertness, decreases fatigue, and decreases reaction time. However, um, ingestion of caffeine does not compensate for sleep deprivation. Effects on sleep and anxiety. Studies have shown that ingestion of more than 200 to 400 mg of caffeine per day induces anxiety by gamma modulation and stimulation of HPA, that is hypoxanthine pituitary axis. Also, uh, variants of adenosine receptors has also shown different effects of caffeine on sleep and the quality, with also difference in metabolism, difference in variants of the cytochrome 450 subfamilies has also shown different effects on the metabolism. However, as expected from effects on fatigue, uh, caffeine consumption later in the day increases sleep latency, and also uh, it can reduce the sleep quality. Now, as we know, uh, after ingestion of caffeine, the urine output is increased. So let's see how. ADH, also known as antidiuretic hormone, it regulates water absorption in the kidneys. It acts on the collecting ducts of the nephron to increase their permeability to water, allowing more water to be reabsorbed back into the bloodstream. Caffeine inhibits the release of ADH from the posterior pituitary gland by blocking adenosine receptors. Adenosine normally inhibits the release of ADH, so caffeine's blockade results in the less inhibition, uh, leading to decreased ADH release. With less ADH present, the collecting ducts become less permeable to water, resulting in reduced water reabsorption and increased water excretion in urine. Not only it inhibits ADH release, it also caffeine also increases GFR or glomerular filtration rate. Um, Caffeine stimulates the release of catecholamines that are adrenaline from adrenal glands. This causes vasoconstriction of the afferent arterioles in the kidneys, which leads to increased renal blood flow and subsequently increased GFR. This increased GFR means that more blood is filtered by the glomerular per unit of time, leading to a higher volume of filtrate being produced in the nephron. This increased filtrate volume ultimately results in higher urine output. Caffeine also inhibits sodium reabsorption in the kidneys, which results in more urine output. Let's break down the inhibition of sodium reabsorption by caffeine. So, caffeine acts as a competitive antagonist at adenosine receptors, particularly adenosine A1 receptors, which are located in the kidneys. Adenosine normally inhibits the release of renin, an enzyme produced by the juxtaglomerular cells in the kidney. By blocking these receptors, caffeine prevents the inhibitory effect of adenosine on renin release. With adenosine receptors blocked, renin release is no longer inhibited. Renin is released into the bloodstream and acts on angiotensin, a precursor protein produced by the liver, converting it to angiotensin 1. This angiotensin 1 is further converted into angiotensin 2 by the angiotensin converting enzyme or ACE, which is present primarily in the lungs. Angiotensin 2 acts on the adrenal cortex to stimulate the release of aldosterone. Aldosterone, which is a steroid hormone, which acts on distal convoluted tubules or DCT and the collecting ducts of the nephron, promotes the reabsorption of sodium ions and water from the tubular fluid back into the bloodstream. Aldosterone also stimulates the secretion of potassium ions and hydrogen ions into the tubular fluid from excretion. Caffeine-mediated inhibition of adenosine results in decreased renin release and subsequent lower aldosterone levels. With reduced aldosterone levels, the reabsorption of sodium ions in the distal convoluted tubules and collecting ducts is impaired. As a result, more sodium remains in the tubular fluid rather than being reabsorbed back into the bloodstream. The presence of sodium ions in the tubular fluid exerts an osmotic effect, drawing water molecule from the interstitial fluid into the tubular lumen. This osmotic gradient created by the sodium retention promotes increased water excretion and leading to diuresis, which is increased urine output. Additionally, the excretion of sodium ions along with the water contributes to the overall increase in urine volume. Caffeine, insulin resistance, and type 2 diabetes. Let's talk about the intracellular signaling pathways involved in insulin action and how caffeine immunosuppression may disrupt them. Insulin binds to its receptor on the cell surface, leading to receptor autophosphorylation and activation of downstream signaling cascades. One of the key pathways that gets activated by insulin receptor signaling is the phosphatidylinositol 3 kinase or PI3K-AKT pathway. The activation of PI3K by insulin leads to the production of phosphatidylinositol 3,4,5 triphosphates or PIP3, which recruits AKT, also known as protein kinase B, to the cell membrane. This PKB or protein kinase B is then phosphorylated and activated by PIP3 bound phosphoinositide dependent kinases and also mTOR or mammalian target of rapamycin complex 2. Activated protein kinase B phosphorylates and regulates various downstream targets involved in glucose metabolism. For example, it leads to the activation of GLUT4 or glucose transporter type 4 translocation to the cell membrane, facilitating glucose uptake into the cell. It also inhibits the glycogen synthase kinase 3, leading to glycogen synthesis. And also it activates a mechanistic target of rapamycin complex or mTORC1, promoting protein synthesis and cell growth. Caffeine is shown to interfere with several components of this pathway, potentially disrupting insulin signaling and glucose metabolism. Studies have demonstrated that caffeine can inhibit AKT phosphorylation and activation, thereby impairing downstream signaling events mediated by protein kinase. Caffeine may also interfere with insulin-induced GLUT4 translocation to the cell membrane, reducing glucose uptake into insulin-sensitive tissues such as muscles and adipose. Additionally, caffeine has reported to activate stress-activated protein kinases such as JNKs or P38 mitogen-activated protein kinase or MAP kinase, which can antagonize insulin signaling and promote insulin resistance. Furthermore, caffeine induced increase in intracellular calcium levels and reactive oxygen species may contribute to insulin resistance by disrupting insulin receptor signaling and promoting inflammatory responses. Furthermore, metabolic studies suggest that caffeine may uh, improve energy balance by reducing appetite and increasing the basal metabolic rate and food-induced thermogenesis, possibly through stimulation of sympathetic nervous system and uncoupling of protein 1 expression in brown adipose tissues. Repeated caffeine intake during the day uh, leads to 5% increase in 24-hour energy expenditure. Limited evidence from randomized study also support a modest beneficial effect of caffeine intake on body fatness. However, caffeinated beverages that are high in calories, such as soft drinks and energy drinks and coffee or tea with added sugar, may lead to excess weight gain. Furthermore, in cohort studies, habitual coffee consumption has been consistently associated with a reduced risk of type 2 diabetes in the dose response relationship, with similar association for caffeinated and decaffeinated coffee. Taken together, these findings suggest that tolerance develops for the adverse effect of caffeine on insulin sensitivity, or that the adverse effect is offset by longer term beneficial effects of non caffeinated coffee components on glucose metabolism, possibly in the liver. Okay, in conclusion, a substantial body of research indicates that the consumption of caffeinated coffee, which serves as the primary source of caffeine intake among adults in the world, is not linked to an increased risk of cardiovascular diseases as well as cancer. On the contrary, the regular intake of 3 to 5 standard cup of coffee per day correlates with a reduced risk of several chronic illnesses, keeping in mind that the coffee intake should be sugar free. It is noteworthy that current evidence does not advocate for caffeine or coffee consumption solely for disease prevention purposes. Nonetheless, for adults without specific health concerns who are neither pregnant nor lactating, moderate consumption of coffee or tea can be incorporated into the balanced lifestyle. So I hope you guys enjoyed this video. I hope you guys learned something new. Please leave a comment if you have any queries. Please like and subscribe to the channel, and I'll see you in the next